Good afternoon, our honorable scientists, forum participants, ladies and gentlemen. Welcome to the flash presentation of Life Science and Medicine. I am Cindy, your MC this afternoon. On behalf of HKLF, thank you for joining us. We are very delighted that we are going to have over 20 young scientists giving a flash presentation in this session. We will invite presenters to come out one by one. Each of you will be given 3 minutes. Please kindly end your presentation when time is up. We have a timer over here for your easy reference. Without further ado, I would like to welcome our first presenter. Everyone, let's give a warm applause to Jiliu Liu.
All right. Hello, everyone. My name is Julio. So my research interest lies in understanding or exploring how cells balance precision and plasticity, two important principles for cells or for our biological system to stay on accurate and adaptable. So during my PhD in Copenhagen, I studied DNA-protein cross-link, a severe form of DNA damage, which basically the protein get trapped or covalently trapped on DNA. So I focus on two enzymes, which is RNF4 and TOPOS. So both of them are sumo-targeted ubiquitin ligase. It means they modified or they target the sumo-modified proteins and tag them for degradation. But interestingly, RNF4 and TOPOS turns out to have strong preferences. For example, RNF4 strongly prefer targeting the polysumo 2,3 chains, and the TOPOS, which I identified as it as a new sumo-targeted ubiquitin ligase, strongly prefer the monosumo 1. Despite these differences, both of them are very efficient, important for the DNA-protein cross-link repair. So recently, I moved to Basel, Switzerland for my postdoc, and also shifting gears from the DNA repair to transcription regulation. So I focus on a group of proteins called small MAF proteins, which are the smallest transcription factors in our cells. So small MAF proteins, they can bind both homodimers and heterodimers. They bind to the same or highly similar DNA motif. One of the key partners is NF2, which is the master regulators of the oxidative stress responses. So basically, the current model suggests that under the normal condition, NF2 get constantly degraded by the KIT1 signaling. But under the stress, the NF2 stabilize and compete with other partners, for example, BAC1 for the DNA binding. But is it the whole story? I suspect not. Otherwise, the competition will be always like this. So I want to see, I aim to understand whether there's additional layers of controls, for example, to fine-tune the precision and to fine-tune how these small transcription factors to bind, like who to bind or when to bind or where to bind. So for example, like this is at least some additional rules. So basically from my PhD, the DNA damage repair to the transcriptional control, I seek to understand how the cells or biological system to have to strike a balance or delicate balance for the precision and the flexibility, which help our cells to be resilient and our life to be adaptable. Thank you.
Thank you, Julian. Wonderful, thank you. Now we may exchange our subject further after the whole flash presentation. Next we have Gonzalo Perez-Rosell for the next presentation. Thank you.
Hello, everybody. So I guess you are all familiar with coral reefs, but you might be not so familiar with what we call mesophotic coral ecosystems. So mesophotic, it refers from the Greek, meso is middle, photic, it's light. So it's these ecosystems that we find from 30 meters all the way down to the limits of the photic zone. You might also be familiar that the shallow coral reefs are disappearing worldwide because of something that we call coral bleaching by global warming. But we don't know much of what's going on underneath at these mesophotic depths, because accessing these mesophotic depths is very difficult, very challenging. We need to use either technical diving or submersibles or this fancy equipment. But every time that we go down, we discover new things for science, which are very interesting. So some of my science and other colleagues, we've been working on this transition zone where we have found a bleaching reduction with depth. We have found unique environmental conditions that translate into unique biodiversity. We have found some of the most coral-rich areas in some, even comparing across the different depths. And also the peak of the richness, which is also the second point. What it means is that in this middle area is the overlap between the shallow communities and the deep communities. So if you want to tackle something like biodiversity loss, you should focus on these depths. However, there is a lot of variability in the natural world. And the reality is that these conclusions are not uniform all across the oceans. For instance, a bleaching reduction seems to be associated with the presence of internal waves. These internal waves will bring deeper, colder, and richer nutrient waters into the shallow areas. And during these heat stress periods, it might mean like a relief of the heat stress. And you can have better outcomes after bleaching with less mortality. Or what we call the deep reef refugia hypothesis, which states that deep corals or deep communities can replace the shallow communities after disturbances. However, this relies on two assumptions. The first one is that the disturbances need to be lower at these deeper depths. We're still finding out. And the second one is that the communities need to be identical, need to be the same, and they also need to be genetically connected. I'm running so much. So in conclusion, my research suggests that when you are studying coral reefs, you need to at least consider these deeper areas, especially this transition zone, because they might be playing key roles in the overall resilience of coral reefs. And the second one is that they are still so understudied. So our curiosity pushes us to go a little bit down because they have unique diversity, they have unique functions, and we need to consider them. They deserve further scientific attention and at least be considered in conservation planning. Thank you so much.
Thank you very much for your presentation. Next, we have Chuan Xin-chong. Please welcome, ladies and gentlemen.
Hi, everyone. I'm glad to be here to share my PhD study in the bone-regulated energy metabolism. I'm Chuan Xin-chong from Hong Kong Baptist University. And we know the obesity has become a big problem in the world now. And in the obesity patients, the bone loss and the glucose handling impairment are two big challenges. So to identify the therapeutic targets to address both complex is very important. So in my study, we identified an enzyme which called 11-beta-HSD1. This enzyme could convert the inactive glucocorticoids to the active glucocorticoids. And we found they are elevated in the obesity patients with bone loss. Then we identified the 11-beta-HSD1 is contribute to the high-fat diet-induced obesity mass with the bone loss and glucose handling impairment. By using the genetic approach, we knock down the 11-beta-HSD1 in the osteoblast of the mass, and we found that the mass could resistant to high-fat diet-induced obesity, bone loss, and glucose handling impairment. So next, we constructed a osteoblast-specific 11-beta-HSD1 inhibitor, and this inhibitor could directly inhibit the HSD1 in the osteoblast of the mass. By using this inhibitor, we found this inhibitor could treat the mass with high-fat diet. The mass didn't show bone loss, obesity, or glucose handling impairment. So in conclusion, we found that blocking 11-beta-HSD1 in osteoblasts could combat obesity, glucose handling impairment, and bone loss. So that's the end of my presentation. Thank you.
Thank you very much for your sharing. For the next presenter, we have Jihan Yang.
Okay, good afternoon, everyone. It's my great pleasure to share you my topic, which is the cancer. I think most of you are very familiar with cancer already. What killed people is not the primary cancer, it's the cancer metastasis. But we are very hard to observe how the cancer metastasis from the primary side to the secondary side. Then we will use the technique called microfluidic. Microfluidic technology is based on the photolithography. We are able to replicate the tumor microenvironment by using a coin-sized chip. So the first chip I introduced here is the confined migration model. In the cancer migration, the confined migration happens everywhere. It's like the blood pressure confined and the fiber confined in the whole cancer environment. So for the first confined migration model, we used the microfluidic device, which is the two-layer device. We are able to create a confinement here. Then we are able to see how the cells will migrate from the side to the other side. Also, we also create these single-cell migration platforms that we are also able to observe how the cell migrate inside of the chip. But it is still on the 2D environment. The 3D environment is more important in the real scenarios. So we also construct this tumor microenvironment model, which is we are able to replicate the tumor microenvironment, which is the hypoxia gradient and the drug gradient, and also different cellular component inside of one chip. Then what are we going to do on the next for using those microfluidic chip, which is the drug screening. So in the drug screening process, we also want to see what kind of drug that able to inhibit the cancer cell migration. So we are able to inhibit the cancer metastasis from the very beginning version. Then we used our device to construct this drug screening device that we are able to screen 36 drugs at a time. And by combining with this real-time microscopy, we are able to see how the cell's behaviors on the treatment of different drug components. And by using this device, we already succeed to find 3 different we see the compound that able to inhibit the cancer metastasis, and also these drugs already, we got the patent granted here. And the second platforms that I'm introducing here is the droplet platforms, because the cancer cells sphere size and the shape matters to the drug screening. Then we are using our smart AI-based sorter. We are able to sort the cancer cell sphere in the certain shape, then perform the drug screening. So by using these platforms, we are able to get a better screening result than more close to the real in vivo scenarios. So that's all for my presentation. Thank you for listening.
Thank you very much for your sharing and insight. Next, we have Xiangfeng Lin.
Hello, everyone. My name is Xiangfeng Lin from Zhejiang University, and I'm an orthopedic surgeon. So today my fresh topic is the plant-derived natural photosynthetic system for osteoarthritis treatment by improving cell anabolism. So the inadequate anabolism is related to most pathological process, for example, aging and injury repair. And the ATP and the NADPH serve as energy currency and the reducing equivalent in our human cells. And in this study, we prepared the nanosarcoid from the spinach, and we used the membrane cell member to encapsulate the nanosarcoid to reduce the immune rejection and choose the closed space application, use the plant-derived photosynthesis to realize the treatment the osteoarthritis by improving the cell ATP and the NADPH and to removing the anabolism of the degenerated chondrocyte in our model. So thank you very much.
Thank you. And for the next presenter, we have Anna Xiaodan Yu.
Hello, everyone. Take a moment and feel your arm or legs. The hard, strong bone underneath is constantly being renewed and maintained by our body. But to stay strong, they need a constant supply of fresh new bone material. Here comes the question is, what keeps this skeletal pool from running dry as we get older? My research tackles this problem by investigating a key gene, MMP14, and its unexpected role in preventing age-related bone loss. We focus on a crucial group of cells, hypertrophic chondrocytes, which act as a major source of these bone-building progenitors in our long bones. Using a genetic mouse model, we specifically delete the MMP14 gene only in the hypertrophic chondrocytes. Our initial findings suggest that adult mice without MMP14 show a severe loss of their trabecular bone. They essentially develop osteoporosis. Why? The mechanism revealed that the pipeline of skeletal stem cell and also the osteoblasts derived from these chondrocytes had completely dried up. This suggests that MMP14 is essential for maintaining this lineage continuum. But that wasn't the whole story. Our multi-omics approach revealed that a second actor, that is the immune system. We found that from our data, we can see there is a significant influx of neutrophil into the bone marrow of our mutant mice. This reveals a new dimension to bone homeostasis. It's not just about the bone cells themselves. MMP14 deficiency doesn't just shut down the bone production line. It also sends a distress signal that recruits neutrophils. And interestingly, our further data suggests that these neutrophils interact with the remaining osteogenic cells, exacerbating the bone loss and hinder their function. In summary, my research revealed that the role of MMP14 in the hypertrophic chondrocyte lineage is a guardian for the bone volume. This will open the new avenues for therapeutic strategies that target this specific cellular crosstalk. I will be happy to discuss you about this fascinating data in my coming paper. Thank you.
Thank you, Anna, for sharing your perspective. Next one, we have Shilan Wang.
Good afternoon, everyone. I'm Shilan Wang from the Chinese University of Hong Kong and also related to the Microbiota Eye Center, which is one of the Hong Kong centers. Some of you may also visit it tomorrow afternoon. So today I would like to share a piece of work did during my PhD period. A brief introduction. The gestational diabetes is defined as the hypoglycemia first recognized during pregnancy. However, how gestational diabetes affected their offspring's gut microbiome are poorly understood. So therefore, in our study, based on a longitudinally prospective birth cohort in Hong Kong, we utilized the metagenomic to profile the gut microbiome of mothers, gut microbiome during pregnancy, and also their offsprings during the first year of life. So our results show that the gestational diabetes mother, their own have distinct gut microbiome when compared to the healthy control, especially characterized with lower gut microbiome diversity and also have some higher abundance of the pathogenesis. And further, we found their offspring's microbiome is fingerprinted by the GDM, especially confounded by the C-section. As we know, the GDM mother, they have a higher proportion of C-section. Therefore, their offsprings may more exposed to the skin or the air microbiome rather than the vaginal or the stool microbiome. Therefore, their offspring's microbiome is changed. And further, we interestingly found the gestational diabetes affect their offspring's head circumference. Especially the male offspring, they will have accelerated head growth. And the head growth is a kind of proxy for the children's neurodevelopment phenomenon. And also, the accelerated head growth have linked to the autism in per study. Then we were interested in what's the potential reason, and then we focused on the gut microbiome with the neuroactive potential, and we found this kind of sex-dependent manner already may happen during pregnancy. Especially we found the mother with male fetus, their gut microbiome have depleted gut premodulars, especially acetate or glutate synthesis. Therefore, this piece of work indicate that the gestational diabetes will affect their offspring's gut microbiome and also link to their later on head growth and neurodevelopment disorder. And So our work are trying to demonstrate that the importance of both prenatal and early life microbiome on the children's health. So further research is definitely worth to follow up their real outcome and also see whether microbiome could help to prevent or mitigate this kind of risk. That's all my sharing. Thank you.
Wonderful. Thank you for your presentation. Next, we have Wong Chen Wei Alex.
Hello, everyone. My name is Alex Wong. I'm currently a postdoctoral fellow at Mass General Cancer Center, Broad Institute, and Harvard Medical School. So in my research at Professor Nia Hakoen and Dr. Moshe's at the Vermans Lab, so what we are trying to work on is the melanoma, which is the most aggressive form of skin cancer. So currently, the standard of care treatment for melanoma is immune checkpoint blockade therapy, which target the negative regulators of T cell activation, including PD-1 and CTLA-4. And we also have the target therapy that target the BRAF and MAP oncogenic driver. However, the response of both treatment is around 40% of patients that will benefit from this therapy, suggesting that there are some driving force that we need to identify some new regulators that can enhance the tumor immunity. So in my project, what we know right now is that melanoma cells could differentiate into more kind of like a neoclass, transitory, and undifferentiated cell state to promote the immune evasion. So recently, what we have done is to use single-cell RNA-seq, and what we have found out is that regardless of the treatment outcome of ICB, what we know is that looks like the patients will exhibit a variety of cell state in terms of their abundance of the tumor. So what we can do is to collaborate with one of the group previously I have worked on in the National Cancer Institute. So what I have found is that if I can use two of the cell lines, which are the melanocytic and undifferentiated. So what I have done later on is that I transduce the GFP and 51.1 into those cell lines, so that we can use flow cytometry to identify these cell lines when we mix them together, so that we can recapitulate the tumor heterogeneity for the patient, that later on we can do some experiment to verify that how the impact of the heterogeneity to drive the tumor immunity. So the experiment recently, what I have done is that I mixed the melanocytic and undifferentiated cell lines together, and in the untreated condition, what I have known is that the melanocytic cell lines looks like they will outgrow the undifferentiated. But if you start to add the interferon gamma or TNF alpha, which are the immune-killing cytokines that we know for sure that they will start to kill the tumor cells. And what we know is that the interferon gamma, TNF alpha, or the combinatorial treatment of both cytokines will actually reduce the abundance of the melanocytic cell line, but not the undifferentiated. And more surprisingly, what we have done later on in the in vivo experiment is that, regardless of the mice with or without the immune system, looks like when you mix the tumor cell lines together, they will outgrow the homogeneous model, which is suggesting that the tumor heterogeneity really can create a more aggressive tumor behavior. So in the future study, what I'm trying to do is to create a 4-cell state model that we are trying to use the surface marker or the GLP labeling technology. And afterward, after we finish the impact of the 4-state model, we'll try to understand how the heterogeneity can drive to the TME modification and also the therapy response. And finally, we'll try to also identify or define the functional role of each cell state in the tumor heterogeneity in vivo. So
thank you, Alex. Wonderful presentation. Next, we have our presenter will be Chi Xiang Pan.
So good afternoon, everyone. There is no doubt that AI has made many achievements in many different fields, especially in protein structure biology. One of the most successful examples is the AlphaFold. But if the AlphaFold can address many different biological questions, so specifically in my topic, I try to study the structural effect on the protein structure. So from the first figures here, you can see that using AlphaFold structure as a replacement of experimental structure to study the change of protein stability, you see that for most of the method, they achieve very similar performance. So AlphaFold are doing a good job in studying protein stability. However, when we talk about some more complicated interactions, such as protein small molecule binding, AlphaFold may not be that perfect as we expected. For example, you can see the two methods listed here. They have some performance deterioration when we try to study the change of protein small molecule binding affinity. During my PhD study, I have been thinking, how can we teach AI better to learn biological representation. And one of the methods proposed in the lab that I stay is the graph-based method. According to the algorithm, we can model the atom as nodes and their interaction as edges. By systematically using different distance cutoff and parameter, we can extract different biologically meaningful features from different depth and different perspective. And it is very intuitive to think that if a mutation happens in such a residue that have many important contact with the surrounding environment, it is likely that to disrupt this important contact and have some strong structural impact to the structure. So the first thing that I did to understand how these changes could lead to disease is I do a case study on the tumor suppressor P53 genes. And you can see that from the statistical analysis, the pathogenic mutation in rats significantly destabilized the protein structure and also the protein-protein interaction. And more importantly, using this graph-based feature, it could also improve the machine learning performance to identify the pathogenic mutation. So combining with the experimental assay, the model performance of MCC can improve from 0.69 to 0.83. So in summary, I think that using this knowledge-based feature could be very useful to addressing different biological questions. And if you want to talk about AI and the application in biology, feel free to come to me and contact me, and also scan the QR code to follow my recent work. My name is Chi-Shen Pan. Thank you.
Thank you very much for your presentation. Next, we have Victor Tam.
Oh, it seems like my slide is in a very different style than others. So my name is Victor. I'm a postdoctoral fellow at CUHK, School of Public Health and Primary Care. So today I would like to share my research interests, two very distinct research interests with my two very distinct roles. So first of all, I'm a radiation therapist at your left side talking about radiation oncology. So radiation therapists may not be familiar with you, but I think in Cantonese or Chinese, 放疗, 电疗. It's more common that you guys may understand. So you may have heard that 电疗, 放疗, radiation therapy may have many side effects. It's like we are actually using high energy X-ray to kill cancer cells. So it's like exposing yourself, part of the region, under sunlight and for a very long time and for many sections. So it's like the patients may have exposed to sunlight and having very severe sunburn for their skin reaction, and may have inflammation at their mouth and also hair loss. So my research interest is to look about the supportive care for the patient who are receiving radiation therapy. So these are some of the work I worked previously in the previous years. First of all, I conducted a trial for my PhD study looking at a new modalities called photobiomodulation, which whether these modalities can relieve these side effects. I also conducted a survey on nasopharyngeal cancer patients, look at their quality of life and toxicities during the survivorship. So in the ongoing project, I shift a little bit to another type of cancer, that's bladder cancer. We try to evaluate the urinary toxicities, that is bladder toxicities, among rectal cancer patients, and we hope to develop a smartphone application to take care of their supportive care needs. So let's come to your right side. Another role of mine is that I'm a basic life support instructor. That is, I teach layperson and also healthcare students about CPR, cardiopulmonary resuscitation, and the use of AED. And one of the points that I tackle is about the willingness to perform basic life support. That imagine people just collapse here. Are you willing to compress the chest and grab the AED? So that's more on health psychology. And previous work I have done a survey on community members, and we found that overall willingness is not that good. And actually, we found that if we refresh their skills every 5 years, it significantly improves their willingness. And this is also based on the theory of planned behaviors. It's actually a behavioral model. So in the future work, we want to identify priority barriers in the bystander minds, why they stopped or hesitate to give support to the patient collapsed here. And we want to develop a culturally adaptive, intention-focused learning model that helps them to boost their willingness. Thank you so much.
Thank you very much for sharing and presentation. Next, we have Kenny Ki Li Lau.
Good afternoon. Tendinopathy refers to persistent and tenderness in a tendon, which is the tough band that connects muscle to bone, and most current treatment mainly relieve the pain rather than address the root cause. The ideal approach is to address what is going wrong inside the tendon so it can truly heal, not just feel better for a while. One major factor linked to tendon health is metabolic syndrome, a cluster of health issues tied to metabolism and inflammation. These issues include conditions like obesity, high blood pressure, and high blood sugar, which often travel together and keep the body in a chronic inflammatory state. These issues, metabolic alterations such as a high body mass index, have been associated with a greater risk of tendon problems, suggesting that metabolism and tendon health are closely related. Further, these metabolic changes can slow tendon repair and even push tendon cells towards dying off, which is why medicine that improve blood sugar, blood fat, and inflammation may reduce tendon problems in the first place. However, no study has yet demonstrated that metabolic drugs can cure tendinopathy, and strong evidence from large representative patient groups remains lacking. That is a gap this work targets to address. The aim of this study is straightforward: to compare how common tendinopathy in people with metabolic syndrome who take metabolic drugs versus those who do not take them. In other words, do patients on these medicines have fewer tendon problems than similar patients who are not on them? To answer that, our plan is to analyze 33 years of medical records from Hong Kong Public Hospital between 1993 and 2025, using the database developed by the Hospital Authority, the organization responsible for all government hospitals in Hong Kong. Patients will be included if they have diagnostic code for metabolic syndrome. The analysis will examine prescription for common metabolic treatments such as diabetes medication, beta blockers, lipid regulation agents, and blood pressure medicine, and will target exposure to tendinopathy at the shoulder, elbow, knee, and foot and ankle. The outcome measure will be the preference, calculated as the number of tendinopathy cases divided by the total number of patients with metabolic syndrome, compared between those who take metabolic drugs and those who are not taking them. We expect that individuals on disease-modifying metabolic therapies will have a lower preference of tendon problems after starting their medicines than those not on these drugs. If confirmed, this finding will support treating metabolic issues proactively to help prevent tendon diseases rather than waiting to treat tendon pain after it appears. And this is the end of my presentation. Thank you.
Thank you very much, Kenny. Next, we have Jamie Zhang.
Hi, everyone. My name is Jamie Chong. I'm a JD candidate at the Chinese University of Hong Kong, and I completed my postdoc fellowship in cardiology at HKU School of Clinical Medicine. So today I would like to present two research projects that I completed during my postdoc fellowship at HKU. So the first one is about the aortic aneurysm. So I look at the risk factor associated with aortic aneurysm, and after follow-up of 3.91 years, I have recorded 468 deaths in 2,186 patients. And then I studied what factors contributed to this all-cause mortality. And I find that after adjusting for the potential confounders, RDW, which is the lab value, is associated with patients with AA male only, so not in female patients. What made my research impactful is that I find that inflammation, anemia, and oxidative stress contribute to the elevated RDW. However, this study is only observational study, so I cannot derive any cause and effect relationship from this study. And also, the data collected are have missing data, so there is no fasting blood result, no obesity indices. Therefore, I would like to start a prospective study looking at what factors associated with AA. And the other study is the pulmonary arterial hypertension. So pulmonary arterial hypertension is the pulmoembolic occlusions of the pulmonary artery, and it has a very high mortality rate. I retrospectively look at the functional class of this patient and then study what factors associated with the all-cause mortality in this group of patients. So after a follow-up time of 4.44 years, 282 patients with PAH were analyzed, and then I find that diabetes coronary arterial disease, anemia, and also and renal disease are actually contributed to the elevated all-cause mortality rate. These are the two pieces of work that I completed during my postdoc fellowship in HKU. However, I think that more analysis have to be done because both of them are retrospective in nature. And then because currently we have a new drug that is called sotatercept, which can be used to treat patients with PAH, and then we will have more papers, review articles and papers talking about the new drug. So that's the very brief analysis that I currently have. Thank you.
Thank you, Jamie. For the next presenter, we have Sheena Ann Hansen Garcia. Ladies and gentlemen, please welcome.
What if the nanomaterials intended to make our lives better can actually pose hidden risk? I am Sheena Garcia, a postdoctoral fellow from the Hong Kong University of Science and Technology, and today I will discuss to you the environmental effects and potential toxicity of amorphous silica nanoparticles. Silica nanoparticles is all around us. We can see them in glass, tires of cars, in cosmetics. In agriculture, silica nanoparticles can enhance plant growth, but their effects vary based on their size and concentration. Our research revealed that small silica nanoparticles of around 10 nanometer can actually inhibit germination of chia seed and growth. So that's in the second figure. While if you increase the size to 50 nanometer and at moderate concentration, which is around 200 ppm, these silica nanoparticles can actually be used to enhance the plant growth and health. We also discovered in our research that these silica nanoparticles can be uptaken not only by the chia plants, but also by the celery plant, here in the first figure, through the method of transpiration. So that is, this nanoparticle can travel from the roots to the leaves and even outside of the leaves of the plant. So studying these, if it can go outside through transpiration, is what is new in the field, because usually the scientists just end with discovering it up to the leaves. So this concerns is that if it goes out of the leaves, then there's a possibility that these silica nanoparticles can be inhaled by humans and can be released through the environment. And lastly, we also checked hollow and solid silica nanoparticles. And we have discovered that the hollow silica nanoparticles pose more concern because they have higher cytotoxicity, and that's probably due to the difference in chemical structure. Hollow silica has these three-membered siloxane rings, which are known to pose hydroxyl radicals, which may be the cause of their toxicity. And lastly, we have also considered having an animal model to further study the toxicity of the nanoparticles, and we have discovered that the hollow silica nanoparticles can cause severe damage into the liver, brain, and kidneys, specifically for the brain vacuolation of the neurons, for the kidney tubular injury, and also ballooning and fatty degeneration. And for the liver, we have the abnormal hepatocytes. So I would like to remind that even if we wanted to promote nanoparticles, we should still balance between the nanoparticle advantages and the possible risk it may cause. Thank you for listening to my presentation.
Thank you for your presentation. For the next presenter, we have Shen Liu. Ladies and gentlemen, please welcome our presenter.
Good afternoon. I'm Liu Xian from PolyU. Glioblastoma GBM is a deadly cancer. A key driver of GBM is the unabiding protein MESH1. Direct detecting it has been challenging due to a lack of the specificity between MESH1 and MESH2 and our incomplete understanding of its regulation. Our work addresses this by uncovering a switch on MESH1 which controls GBM source behaviors. So what did we find? we discovered an oncogenic switch of phosphorylation at serine 347 site located in the previously poorly understood C-terminal methylation. Why is this important? Clinically, we find phosphorylated methylation at serine 347 is significantly elevated in patient tumors and associated with poor survival in animal models. Functionally, this single switch acts as a master regulator. 347 phosphorylation promotes cell invasion, tumor initiation, and reorganize the cytoskeleton. It also suppresses cellular senescence, allowing tumor cells to escape the growth arrest with a low P21 level. In essence, this one switch coordinately drives malignant tumors. Knowing this, we asked, can we block this switch? We designed a decoy peptide that mimics the 347 site. This decoy peptide acts as a competitive inhibitor, suppress natural phosphorylation of methylation. A key advantage is the specificity. Applying this decoy peptide effectively reduces tumor formation, invasiveness, and associated the cytoskeleton changes. In conclusion, our work identified phosphorylated methylation at serine 347 site as a previously unknown druggable target, which controls GBM metastasis, suppress senescence. Targeting this switch with a specific decoy peptide, we could suppress these malignant features. Our future work will achieve this decoy peptide into a real therapy by testing it test the PECT in other GBM hallmarks and developing strategies to deliver it across the blood-brain barrier. That's all. Thank you.
Thank you very much for sharing your insight with us. For the next presenter, we have Ho Chi Ching Vivian.
All right, this is probably the only slide right now I show people. So AI, everyone is talking about AI right now, but have you ever tried to use AI to manage your health and social needs? I'm Vivian Hui from PolyU School of Nursing Center for Smart Health. I'm going to show you a couple of chatbots that we developed to train an AI to be emotional supportive. The first one we're working with is actually a nurse bot. So in nursing practice, we have a lot of routine that we actually have to follow guidelines. But the guideline could change every single day. So we've tried to reach out to more nurses to equip them with more like standardized policies. So we used retrieval augmented generation with the large language models, and then we built some knowledge graph to understand different guidelines, latest release, and also match with the information needs by the nurses. So we have lots of focus group to get into the patients and all the stakeholders. So we build a nurse board to facilitate their workflow. The second one is a domestic violence chatbot. I'm not sure whether any one of you encounter people suffering from domestic violence. It's very suffering because it's a long-term consequences causing a life-threatening mental health issues, and you could be suffering from like stigma, embarrassed to tell people. They are very, very isolated. So we developed a chatbot to see how we can actually train them as a nurse to encourage them to seek help, identify the help-seeking barriers, as well as connect them back to the community. So we are also trying to connect with the NGOs. That means we have a lot of pictures working with the NGOs to show them what kinds of services we can connect them. And so we developed agentic AI to connect them to different services in the community so that they can seek help easier. Another one is for neurobot. A lot of patients, when they go into the hospital suffering from, they are waiting for the neurosurgery, it's very complicated, complex, and they're very stressed about it. So we analyzed the patient's needs and also the routine culture of people, what questions they ask the most. We also use the standard guidelines as well as the most asked questions by using like topic modeling or sentiment analysis to build a knowledge graph to understand this patient's condition and map them with their cancer trajectories to feed them some relevant information and to reduce their anxiety level. So So all these type of chatbots were all developed. The most recent one is the e-mobile. You can see it from the right-hand side. So we are going to track the mood and the stress trends to showing that how their mood tracks, how they move on their mood stress, and also they can also look at their high-risk alert because we developed some triage model, like we triage the cases from the hospital in A&E, and telling them how is their risk score, what kinds of risk factors you're suffering from, and how we can connect them to the community as well. So we are actually trying to develop these chatbots not only to helping the patients, but also by training our healthcare professionals, because the AI can actually get a lot of data for our in-house training. Originally, we might need to deal with the patients in person, but now we can use AI to get the retrospective study much easier. Thank you.
Thank you, Vivian, for your presentation. Next, we have Jingyi Luo for her presentation. Let's welcome.
Hello everyone, my name is Luo Jinyi. I'm the PhD student from the University of Hong Kong. Today I'm going to talking about a unique post-translational modification on microtubule. It is the acetylation of the K40 residues. It's special because it's the only known modification that happens inside the microtubule numer. But however, how the tubulin acetyltransferase could access into the internal site across the micrometer non-microtubule remains unknown. To answer these questions, we use a cryo-EM. We determine the structure of the tubulin acetyltransferase in complex with microtubules. This revealed the tubulin acetyltransferase could have multi-interaction with their psychological substrate. One important finding is that the tail of this enzyme could anchor at the hydrophobic taxane binding pocket on the beta tubulin. It is very interesting because it means that the very famous cancer drug, pantitaxol, which also targeted this pocket, could compete with our enzyme. So combine this structure inside and the following mutagenous and the single molecular experiment allow us to bring up a model for the tubulin acetyltransferase to assess, recognize, and modify the residue in the microtubule lumen. First, the microtubule like to employ the acetyl-CoA bonded acetyltransferase into the microtubule outer surface, and the tail, the disordered negative charged tail, helps infiltrate into the lumen and anchors at the taxane binding pocket. And this anchor increases the possibility of the whole enzyme to go to pass through the letters and forms the ternary complex with unisolated K40 residues. And once the acylation happened, the CoA could dissociate from the enzyme, and the APRO, which decrease the affinity of the enzyme to the microtubule inner lumen. And the APRO enzyme could dissociate from the lumen and then loaded another acetyl-CoA to form prepare for another cycle of acetylation. In all, the alternated high affinity acetyl-CoA bond state and the mobile APRO state allows the acetyltransferase to isolate the alpha tubulin over the microtubule, micrometer long microtubule lumen. And that's all. Thank you.
Great to hear your sharing. Thank you very much. Next, we have Yuehua Tang.
Good afternoon, everyone. My name is Yuehua. I am a postdoc at the University of Hong Kong and a big company in Science Park. Today, I'm going to present my research on leveraging graph convolutional networks for multi-component crystal prediction and classification. Let's start with the MCC background. Multi-component crystals, or MCCs, are crystalline materials composed of one active pharmaceutical ingredient, API, one conformer, and optionally several molecules. They can significantly enhance the physicochemical properties of pharmaceutical drugs, like stability and solubility. However, screening suitable conformers to form stable MCCs is still a big challenge. So in our work, we aim to build an AI model for MCC classification, which can distinguish between different types of MCCs, like co-crystal, source, and source. To the best of our knowledge, this is the first AI model for MCC 4-class classification. We use the graph convolutional networks which can learn directly from the molecular structures and crystal patterns of MCC crystals from the database. At the same time, we also use other traditional prediction methods like 3 knowledge-based methods and 2 quantum chemistry methods for comparison. We train our GCM model and then experimentally validate it using 2 APIs, cortisol and coprol, these 2 molecules for hair loss, with 32 conformers with carboxylic acid groups. Our model showed a high accuracy in MCC classification and prediction overperforming other traditional prediction methods and benchmark models. And our GCM model can also provide formation interpretation for MCC formation like protein transfer in source, hydrogen bonds in co-crystals, and other intermolecular interactions contributing to MCC formation. So in conclusion, our GCM model can provide guidance for MCC design and pharmaceutical development. Thank you for your time and attention. Thank you.
Thank you for your presentation. For our next presenter, we have Shi Wenlong.
Hello, everyone. I'm Shi Wenlong from Karolinska Institute. As you may all know, viruses are parasites inside the host cells. They completely rely on the host cell machinery to successfully replicate. So this topic, I will introduce the battle between viruses and host cells to try to figure out what we can learn from it. Here we use a SARS-CoV-2 infection model, and as you can see, once the viruses enter the host cells, some stress sensors can be activated, like PKR and the PERK, and then leads to the formation or assembly of these so-called stress granules. This process is considered to be antiviral because it can not only inhibit the viral protein translation, but also induce the interferon signaling pathway. However, the viruses has evolved countermeasures to combat this process. As the SARS-CoV-2 nuclear capsid protein and protein accumulated, they could hijack the core component of the stress granules, G3BP, the green protein, and then leads to the disassembly of these stress granules. And more interestingly, we observed the phase separation of M protein and G3BP, and then they could be recruited to the double membrane vesicles, which is the replication organelle of many RNA viruses. And then they concentrated near the double membrane vesicles, then recruit the 40S ribosome subunit to facilitate the viral protein translation. We observe a condensed ribosome accumulation near the DMV through transmission electron microscopy, and also enhanced the translation efficiency through confocal microscopy. And of course, the phase separation of G3BP and N could facilitate the virus particle assembly. And there was one paper came out last year. They can actually detect the G3BP protein inside the purified virus particles. So what we can learn from it, we looked into the crystal structure of this complex in detail, and we just mutated 2 amino acids, which could block the interaction of G3BP and N, and it turns out the viral mutant is so attenuated both in vitro and in vivo. So yeah, thank you for your listening.
Thank you very much for sharing and insights. For the next presenter, we have Chen Yao-Tun.
Good afternoon, everyone. Today, I would like to share with you my research related to the anti-tumor effect of melatonin on the pancreatic cancer. So in the preclinical model, we found that the melatonin has an anti-tumor effect in the mice, but not directly on the cancer cell. So we hypothesized that the melatonin may have an effect on the tumor microenvironment instead of directly on the cancer cell. So later on, we found that the melatonin can increase the neutrophil like population into the tumor microenvironment, which are further characterized as the anti-tumor N1-like neutrophils by functional assays. So by depletion of the neutrophils, we found that the anti-tumor effects of the melatonin are offset. So we further look onto how the melatonin has the signaling effect on the neutrophils, and we found that the melatonin induced the cancer cell to secrete the chemokines called the CXCL2 to recruit the anti-tumor neutrophils into the TME, and in which the neutrophils will also exert an anti-tumor function called the neutrophil extracellular trap, which is a web-like structure which kills the cancer cells by direct cell-to-cell contact. And for further, we perform a proteomics analysis, and we found that the tumor cells are induced by the melatonin to promote the CXCL2, which leads to the upregulation of the fatty acid oxidation in the neutrophils. And this upregulation of the FAO can promote the latosis, which is the net formation of the neutrophils, and further promotes the anti-tumor effect. And we finally to correlate these preclinical findings in our clinical samples, and we found that in the patients, we observed a higher, when the patient express a higher melatonin level, they also show a higher CXCL2, higher neutrophil infiltration, and higher netosis formation in their pancreatic cancer tissues. So we suggest here that the melatonin has a very important role in the regulation of the tumor microenvironment, and the supplementation of the melatonin as a natural supplement, health supplement, can also has a potential role of the anti-cancer effects of the pancreatic cancer. Thank you.
Thank you for sharing your research with us. For the next presenter, we have Tan Yi Ding.
Good afternoon, everyone. I'm Tian Yiding, a postdoctoral researcher from Tongji University in Shanghai. Sometimes I'm wondering what would happen if the world around us were in total chaos. Probably nothing would work. Traffic, weather, or conversation may all collapse into noise. So the same is true in the nucleus of the eukaryotic cell. DNA is not a loose string. It is intricately condensed, folded, and compacted into a precise three-dimensional architecture, maybe from chromatin territories to AB compartments and topologically associated domain and chromatin architecture loops. So it is a regulatory code inside the nucleus. It may contact the distal enhancer or suppressor to the gene promoters. So when this kind of 3D genomics is disrupted, maybe consequence can be profound. So my work asks the focused question is that how multidimensional chromatin architecture regulates tumorigenesis, especially in orphan cancers such as uveal melanoma, where the early diagnosis and treatment options are limited. Over the last decade, advances in 3D genomics have moved us to study how spatial genome changes underlie cancer biology. For example, maybe aberrant chromatin architecture could lead to oncogenes next to inappropriate enhancers and to alter epigenetic marks or silence tumor suppressors. All of this maybe could drive tumorigenesis. In short, multidimensional chromatin architecture is a hidden layer of oncogenic control. Different to genetic controls, it is a kind of epigenetic layer. So by reading and rewiring that layer, maybe we could provide a promising strategy to conventional therapies for tumor. So that's all. Thank you very much.
Thank you very much for your sharing. For the next presenter, we have Choi Chi Yen Juliana.
Hello. Imagine you haven't eaten since yesterday. Would you rather have a sweet but 0-calorie jelly, or would you pick a nutritious banana? If you pick the banana, you probably think like my flies do in lab. Congratulations. Hello, I'm Juliana Choi. I am a DPhil candidate at the University of Oxford, studying how fruit flies learn and remember things. So, fruit flies, you might commonly see them in your kitchen. But turns out their attraction to really sweet things and their conserved behaviors make them an ideal model organism for understanding the molecular mechanisms for these complex behaviors. So the fruit fly brain here is composed of roughly 140,000 neurons, and they are the size of roughly a poppy seed. Around 2,000 of these neurons comprise what is known as the mushroom body, the center for learning and memory. And we train fruit flies to learn and remember things similar to Pavlov's dog. But instead of a bell and food, we use smell and a sugar reward. We train them to associate these things and see whether or not they remember this immediately after training, or whether or not this memory is consolidated into long-term memory. And turns out, dopaminergic neurons that innervate the mushroom body are essential for driving flies to either approach or avoid a stimulus. Now, what do I work on specifically? I hope, in my PhD, I have looked at how the map of all the brain's connections and the genes that they express combine to help us understand how learning and memory is regulated in the brain. Specifically, I have found that different populations of dopaminergic neurons are necessary for regulating distinct time phases. What does that mean? Turns out, as few as 7 neurons per hemisphere is sufficient to completely regulate long-term memory. Imagine that. Additionally, using the transcriptome, so which genes are expressed where, has allowed me to identify key neuromodulatory receptors that has demonstrated that they are able to regulate learning and memory in a hunger state dependent manner. So as you might imagine, if you were hungry, you'd pick the banana, but if you were really full, the jelly is a really nice treat. And with that, I hope to understand the underlying molecular mechanisms and genetic underpinnings of how complex behaviors like learning and memory work. Thank you very much.
Thank you for sharing your findings with us. For the next speaker, we have Kai Cheng Chu.
Good afternoon, everyone. I'm Kai Cheng Chu from HKUST. My current research focuses on the theoretical studies in phase separation dynamics. You can see that from the figures shown here, during the phase separation dynamics, it describes the aggregation and assembly of biomolecules. As shown in the top right figure, the nucleation process involves an intermediate nucleation barrier, which corresponding to a critical nucleation size. Based on this size, the overall phase separation dynamics is separated into 2 stages, the early and the late stages. Since the early nucleation stage is not favored by the thermodynamic driving force, it is a rate limiting step in phase separation. However, a very important aspect has been missed in previous studies. Biochemical reactions, such as protein phosphorylation, would alter hydrogen bond and van der Waals interactions inside the condensed clusters. To describe this reaction-mediated mechanism, we design a phenomenological description of for the chemical states as an ordinal parameter based on their theoretical methodology in statistic physics. We conducted non-equilibrium simulations and found that the heterogeneous structural patterns naturally emerge, as shown in the figure here. Besides, we see that the structures were not static. They dynamically evolved while the clusters continues to grow with time. Furthermore, we also analyzed how the reaction will change the nucleation rate. As shown in the bottom figure here, the relationship between the nucleation rate and the reactions are determined by the interaction strength inside of the condensed clusters. At this moment, we haven't applied this model for specific biological systems. So if you are familiar with any particular system that can be experimentally tested, we really like to hear your thoughts. Thank you.
Last but not least, our last speaker would be Michael Rivera.
Don't worry, everyone. This is a real plastic skull. So I'm Hong Kong's first person ever to get a bachelor's, master's, and PhD in biological anthropology. Biological anthropologists study human evolution and the 6 million year long story that we took to reach where we are today. So I study why is it that we walk on 2 legs, develop big brains, have the capacity for language and art, and to cook our food. And I do a lot of different research all around the world on this topic. So for example, I think over my long career, I've looked at over 4,000 or 5,000 fossils and skeletons, skeletal individuals from around the world, from all over the place and all time periods. The oldest one I've ever held in my hand is about 2 million years old, this cranium piece in South Africa. I also am involved in a lot of projects in Southeast Asia and East Asia where we look at archaeological skeletons of Homo sapiens, and we consider how is it that they can give us details about health and disease in the more recent past. Humans have undergone a lot of changes in the last 10 or 12,000 years only, and it's because we're so successful at going into all kinds of environments, and somehow I want to untangle like which aspects of the skeletons of our bones and teeth come from genetic influence, come from environmental influence, or maybe the choices we make in our diet and our lifestyle. I hold the keys to my lab here. This is the Hong Kong U skeletal collection. There are 370 individuals there that date to about the 70s or 80s, and I'm in charge of doing research about who they were and what they went through. These are the 3 foundations of my job, and I think it's so important to share what we do in science with public audiences and with our youth. So for example, 20 of these undergrads came with me to the Bone Lab this summer to work as interns, and they all submitted these abstracts to the biggest conference in my field, and we're all going to go to the US next year to present on our studies of reactive osteopathy, of arthritis, of malaria, dental disease. We find all of these instances in our bone collection. I wouldn't be here if I wasn't inspired by a TV show in the States. There's a show called Bones, and it aired for 13 years, and it inspired me to do what I do. If there wasn't a lot of outreach, I wouldn't be here. So I think it's so important to do the same myself. And I give a lot of talks in museums and in primary schools and secondary schools. I think that it's very important because I tell the stories of people who can no longer tell their own story. And if you think about the chairs that you're sitting on, if you think about spacesuits being designed, or you think about how COVID-19 was a big challenge for us, anthropologists were probably involved in the solutions. Thank you so much, everyone.
Thank you very much for your sharing and being the last presenter today. Everyone, to close this session, I would like to invite everyone to give a big round of applause to all our presenters. We have listened to 23 very innovative and inspiring ideas. I believe you might have found your interest in some presented subjects, or in some way you were inspired with the new ideas. Thank you very much, all the presenters. For our forum participants who enroll in the coming session, the flash presentation, or if you are one of the speakers of this session, please proceed to the reception counter for check-in. If you would like to review the list of presenters and topics, you can scan the QR code on the screen or at the LED wall in the foyer. For those who are not attending the next session, but joining us for the dinner, we have arranged a coach to transport everyone to the peak. Please arrive at the coach boarding area at boarding tent before 5:45 p.m. At 4 p.m., we will continue the next round of flash presentation. This room will continue to focus on life science and medicine, while the other room will focus on astronomy and mathematical sciences. Everyone, thank you very much and see you soon.